All right, hello, this is Hunter, and this is content review for Biology 211. Today we're doing cell signaling and the cell cycle. So how are chemical signals translated into cellular responses? I'm just going to read this off. Um, we'll get into the worksheet here in a second. So G protein linked receptors, receptor tyrosine kinase, and ion channel receptors are plasma membrane proteins. So these are proteins embedded in the plasma membrane. Protein receptors found in the cytoplasm or nucleus of the cell are the fourth type, and some signals, for example, a protein hormone, interact with signal receptors in the cell membrane to initiate the process of signal transduction. This often involves changes in a series of different relay molecules in a signal transduction pathway, and ultimately the transduced signal initiates an intracellular response. So a signal molecule from outside reacts with this transmembrane protein and is transformed into some other kind of signal, which initiates a response inside the cell, intracellular. Other types of signals, for example, steroid hormones, can diffuse through the cell membrane and interact with intracellular receptors. For example, testosterone interacts with its receptor in the cell's cytoplasm, enters the nucleus, and causes the transcription of different genes. So these signal transduction pathways are important because some of the signaling molecules are not lipid soluble. They cannot pass that lipid membrane on their own. And so we use these receptor proteins. So consider the two signaling systems and their associated vocabulary below. G protein receptor system and a receptor tyrosine kinase system. How are these similar? We're going to look at both the structural similarities and similarities in how the systems function. Right, so both. In both systems, make this as big as possible, keep my handwriting as tidy as possible for y'all. In both systems, the receptor proteins are bound in the cell membrane. Receptor is membrane bound. Okay, so the binding of a signal molecule to the receptor activates them. In both, activated by signal molecule binding to the receptor. And the role of that activation or that activated relay protein is to activate other proteins to produce a cellular response. So this results in activating other proteins and an intracellular response. Question 2, how are the two systems different? Consider both the structural differences and differences in how these systems function. All right. So the G protein linked receptor is one of my favorites. It's the adrenaline is your sort of model signaling molecule here. And so the G protein linked receptor protein is a single unit that becomes functional when activated by its signal molecule. And the tyrosine kinase receptor proteins, it's two proteins that must be activated by signal molecules and then aggregate to become activated. So how do I want to organize this information here? Let's just split it down the middle. We've got G protein, and we've got TK receptor on the right. Let's see here. The G protein unit is activated by the signaling molecule, and this results in a conformational change. releases a GDP. Well, converts an associated GDP to GTP and releases it. And then the activated G protein moves along the membrane and activates a specific membrane-bound enzyme, which produces the cellular response. So the tyrosine kinase receptor, it must also be activated by a signal molecule, but then they aggregate to come together. So activation results in aggregation, so the proteins come together. And then this activates a number of different responses within the cell. These activated relay proteins are not membrane bound, it should be noted. Relay proteins not membrane bound. So a question in the chat, does it actively replace GDP or does the G protein do that itself? I want to make sure that I get the language here exactly right. So the G protein complex has a GDP, guanine diphosphate, bound to it in its inactive form. When the receptor protein changes conformation, when that signal molecule binds to it, that allows for, excuse me, no, I'm sorry. When the receptor is activated, a GTP from the cytosol can then bind to that inactive G protein to activate it. It loses one of those inorganic phosphates, and then that allows for the movement of that G protein, part of that G protein complex, onto the next protein, which is the relay protein, which is membrane bound nearby. So does it actively replace GDP or does the G protein do that itself? The GDP is a result of the GTP from within the cytosol binding to the activated receptor molecule, I believe. I hope that answered your question. No problem. Alrighty. Unfortunately, our sheet got a little split here on the printing. So both systems can generate elaborate multi-step signal transduction pathways. These pathways can greatly amplify the cell's response to a signal. The more steps in the pathway, the greater the what? What is this supposed to say? The more steps in the pathway, the greater the amplification of the signal is what that's supposed to say. Explain how this amplification can occur. Let's see, other system, another mention in the chat. Let me read here for a second. Oh, thank you, copying and paste. Appreciate you. I have another copy here. For some reason, the printing into this document gets a little weird sometimes. So in a signal transduction pathway, each activated enzyme or second messenger has the potential to catalyze more than one reaction. So let's in a signal transduction pathway, each activated enzymes are usually proteins, or second messenger, has the potential to catalyze more than one reaction. This is why that signal transduction is so important for increasing the strength of these signals, and they really can snowball out of control really quickly, um, which is a topic for a different lecture. So each of its reaction products similarly has the potential to trigger the more than one reaction. Each reaction product has potential to catalyze more than one reaction. Resulting in signal amplification. Big time. All right. If you have any questions about signal transduction, drop it in the chat. I will answer what I can here. We're going to move on to the cell cycle. So what is mitosis? What is the overall purpose of mitosis? I'm going to draw a little doodle here. The overall purpose of mitosis is to produce daughter cells that are identical to the parent cell. Daughter cells that are identical in every way to the parent cell. This will be important. So to do this, the cells must first duplicate all of their chromosomes, and then the chromosomes must be equally divided among the daughter cells such that each has the same complement number and kind of chromosomes as the parent cell. So keeping that in mind, let's look at our next exercise. So the fruit fly has a total of 8 chromosomes, 4 pairs, in each of its somatic cells. Somatic cells are all cells of the body except those that will divide to form gametes, ova or sperm. And let's review the events that occur in the various stages of mitosis. So keep in mind that the stages of cell division were first recognized from an examination of fixed slides of tissue undergoing division. On fixed slides, cells are captured or frozen at particular points in the division cycle using these static slides. Early microscopists identified specific arrangements or patterns of chromosomes that occurred at various stages of the cycle and gave these stages names, interphase, prophase, and so on. Later work using time-lapse photography made it clear that mitosis is a continuous process. Once division begins, the chromosomes move fluidly from one phase to the next. All right, so assuming you are a microscopist viewing fruit fly cells that are undergoing mitosis, within each of the circles on the following page, draw what you would expect to see if you were looking at a cell in a stage of mitosis indicated. So this one's a little funny because they've actually already drawn this for you. So what we're going to do is we are going to highlight those changes that are most noteworthy as we go from each step here in mitosis. So what's important about prophase? The steps of mitosis have begun. The first thing that we're going to notice is that this nuclear envelope starts to dissolve. That nuclear envelope, remember, is holding all of the genetic material within the cell inside the nucleus. And here we see that they've already consolidated into these chromosomes. So in prometaphase, our next step here, all we can really say here is we can see these spindles starting to form, right? And these spindles are starting to exert force on these chromosomes. Starting to exert force on these chromosomes. And these chromosomes, one second here. These chromosomes start to align on the metaphase plate. That metaphase plate appears right over here. So in metaphase, our chromosomes have aligned at the equator. And then anaphase is where the exciting stuff starts to happen. This is where the division becomes really apparent, right? Our chromosomes split and become sister chromosomes. And they're drawn towards opposite poles. Telophase, our nuclear envelopes start to reform. And at cytokinesis, we get this new division that divides the cytoplasm. From the parent cell. So division of cytoplasm happens between daughter cells. And then we have two identical daughter cells in interphase. Interphase is the space between divisions. All right, so what are our products of mitosis? How many cells are produced at the end of a single mitotic division? Single mitotic division. 2 cells. How many kinds of cells are produced at the end of a single mitotic division? Remember, this is identical to the parent cell, so we get only one. Only one type of cell is produced. The two daughter cells are produced, but they are identical to each other, to the parent cell that gave rise to them. Remember that that parent cell no longer exists, right? We began with only one. Two is what we then become. All right, 6 centromeres. 6 centromeres are observed in a prophase cell from another species of insect. How many pairs of chromosomes does this organism contain? How many pairs of chromosomes? So if we're in prophase, let's look back up. If we're in prophase, our chromosomes have consolidated into that nice X shape, right? we have 6 centromeres. That gives us, let's see, we've got number of centromeres visible per cell. Okay, so they gave us the answer in the prompt. How many pairs of chromosomes does this organism contain? I'm sorry. This organism has 3 pairs of chromosomes. So remember, we've got, actually, how do I want to? Yeah, we'll come back to that. Okay. This organism has 3 pair of chromosomes. For each stage of mitosis, indicate the number of centromeres you would expect to find and the number of copies of chromosomes attached to each centromere. In prophase, or beginning division, we have 6 centromeres visible per cell, and the number of chromosome copies attached to each of those centromeres would be 2. So we have a centromere, copy of our chromosome. So in anaphase, remember, is towards the end of our cell division. Anaphase, right here, we've split those. So the number of centromeres visible per cell, we're up to 12. Started with 6, we get up to 12. And each centromere has 1 chromosome copy attached to it. So we've talked through mitosis. Meiosis is not mitosis. I've heard some folks say meiosis instead of meiosis. It makes it a little bit easier to remember that they are distinct from each other. The overall purposes of meiosis, the overall purpose of meiosis is to produce the diploid chromosome, to reduce the diploid chromosome number by half. Reduce the number of chromosomes by half from diploid to haploid. This is a very specific half of the chromosome. The haploid cell contains 1 member of every pair of chromosomes found in the diploid parent cell. The haploid cell contains 1 member of every pair of chromosomes from the diploid parent cell. In what types of organisms does meiosis occur? Meiosis occurs in all eukaryotic cells that will produce gametes, ova or sperm. So this is where the evolutionary advantage of sexual reproduction really shines, because we're going to get a greater degree of biodiversity through meiosis, through um, meiosis than we do through meiotic division compared to mitotic division. And we're going to get into a little more detail in that here shortly. All right, so the fruit fly has a total of 8 chromosomes, or 4 pairs, on each of its somatic cells. This is basically the same prompt. So we're going to continue on to the illustration that they've already given us. Meiosis. Meiosis 1 and meiosis 2. They kind of gave it away with the Roman numerals here, but I just wanted to make that a little bit clearer. And again, we're going left to right, top to bottom. Do you need to do the illustrations if it's already present? Absolutely not. However, in memorizing these, especially the differences between them, tracking the number of chromosomes, things like that, I found it extremely helpful for my own edification to redraw these over and over and over again, and that was my way of memorizing these steps and the important features of them. So no, for the purposes of getting that sweet extra credit, you do not need to redraw these, but it would behoove you to, if only to support your learning of the material. All right. Let's look at our important features of meiosis 1. We have a lot of the same features that we saw in mitosis. We've got that dissolving of the nuclear envelope, but importantly, what's unique to meiosis is these tetrads form, and this is where crossing over occurs. And this is the real moneymaker in terms of genetic diversity that results from sexual reproduction, because you have material from parent 1 and material from parent 2 coming together and swapping the part of the chromosome that they're on. Chemically, this is really cool. We don't have time to get really deep into that, but it's a very, very cool process. Let's see. Cool. That's prophase. Prometaphase 1. Uh, is this pen thicker than? Yes, it is. Nuclear envelope dissolves. Here our tetrads line up again on that metaphase plate. Tetrads split in anaphase 1. Now, again, this is where that magic happens, because you've got genetic material. Let me go back here. I'm going to try and use my color coding here. You've got genetic material from each parent that is starting to swap. So you get these hybrid chromosomes that result in completely genetically novel combinations, distinct from their parents. Telophase 1, your nuclear envelope reforms, and cytokinesis. This is the beginning of the formation of your new daughter cells and the division of that cytosol. Excuse me. All right, meiosis 2 should look really familiar because it mimics mitosis really closely. So prophase 2, again, you've got your dissolving of the nuclear envelope, this formation of that spindle architecture around the edges here. Prometaphase 2, your nuclear envelope is dissolved and those spindles are starting to exert forces on the chromosomes. They align with the metaphase plate. Anaphase 2, those chromosomes get pulled apart and start to move towards the poles. Telophase, you have the reforming of those nuclear envelopes. And finally, cytokinesis, the division of the cytoplasm and the formation of two new daughter cells. So there was some discussion over whether this was an error, whether there should be two additional daughter cells formed from this. I think what we have to remember, at least for the purposes of this diagram, is only one of these daughter cells is represented here in meiosis 2. And so from one of these daughter cells, you get an additional two daughter cells at the end of meiosis 2. But really, if we start with n number, well, you can't see that. If we start with n number of parent cells, at the end of meiosis 2, we have 4n. 4 times the number of cells that arise from that parent cell. I hope that makes sense. All right, so now we're going to kind of rehash what we just said. So consider a single cell going through meiosis. How many cells are produced at the end of meiosis? So one single cell produces 4 daughter cells. How many chromosomes and which chromosomes does each of the daughter cells contain? So each daughter cell is going to have half the number of chromosomes from the parent cell. So each daughter cell has 1/2 number of chromosomes. So that's the first part of the question, how many? Which chromosomes? Each daughter cell contains one member of each pair of chromosomes found in the parent cell. Contains one member of each pair of chromosomes from parent cell. Six centromeres are observed in a prophase 1 cell from another species of insect. How many pairs of chromosomes does this organism contain? So we're going all the way back. Prophase 1, if we have 6 centromeres, that gives us 3 pairs. 3 pairs, remember centromere. 1, 2. Okay. Excuse me. I have fallen prey to the exact vocabulary trap that you should never fall for, because that will hurt your grade. Don't do that. Know the difference between a centrosome and a centromere. All right, so for each stage of meiosis, indicate the number of centromeres you would expect to find and the number of copies of chromosomes attached to each centromere. In anaphase 1, things are starting to move apart. You should have 6 centromeres visible per cell, and the number of chromosome copies attached to each of those centromeres would be 2. just to visualize this here, we're talking about anaphase 1, right here. So in this particular illustration, we have 1, 2, 3, 4, 5, 6, 7, 8 centromeres, one pair per each of those. Or, excuse me, two pair. Number of chromosome copies attached to each centromere. Prophase 2, we've got 3 centromeres visible per cell for these 3 pairs. And now, it's new, we have 2 chromosome copies attached to each centromere after prophase 2. Prophase 2 here. Let's go back and look at our diagram. No, that makes sense. That makes sense. Okay. Drawing attention to a contrast that doesn't exist. I apologize. All right, so non-disjunction. I'm going to highlight this in green just because it's a new word. Non-disjunction, the prefix non meaning not, disjunction, coming apart. Or separating. Non-disjunction of sex chromosomes during human gamete formation may lead to individuals with sex chromosome trisomy, trisomy having 3 chromosomes. An individual with the sex chromosome trisomy of XXY may have resulted from non-disjunction occurring in, now this is a prompt here, true or false. Could trisomy occur if non-disjunction occurred in meiosis 1 in the father's sperm production? So production of gametes, meiosis 1. Let's look back up here. Meiosis 1, if we've got our male cell here, if it's X and Y, those stayed together. We have daughter cells with both or none. So this could be true. Meiosis 1 in sperm production would result in some gametes with both an X and a Y, and some with neither X or a Y. Some with both XY, some with neither. If an XY sperm fertilized an egg carrying an X chromosome, then an XXY individual would be produced. So could we get this non-disjunction in meiosis 2 in the father's sperm production? This would be false. So meiosis 2, we're looking up here. Let me see if I can. How to explain this a little bit better. Yeah. I don't think so. Okay. In this case, non-disjunction during meiosis 2 of sperm production would produce sperm with either 2 X's, 2 Y's, or no X or Y. One of any of these sperm fertilizing an egg with an X chromosome would not produce an XXY individual. Number 3, if it occurred in meiosis 1 in the mother's egg production, yes. Similar story here. So non-disjunction in meiosis 1 of egg production could produce eggs with 2 X's or no X's. I'm just going to do a dash for nothing. Um, and if fertilized by a sperm carrying a Y chromosome, then an XXY individual would be produced. So XX plus Y. Again, this is your egg cell. Gives you XXY, you get your trisomy. And finally, meiosis 2 in the mother's egg production. So this one's actually true. So if we got non-disjunction in meiosis 2 of egg production, um, we could produce eggs with two X's or no X's again, because again, we're only working with X's in these egg cells. Cool. How do mitosis and meiosis differ? Go back, review these, spend some time with them, redraw them. And then fill in the chart. So interphase, the phase between divisions, between phase, between phases. This phase is identical for mitosis and meiosis 1. Identical. I don't know why I'm using such an eclectic mix of capital letters and not. Similarities, they're identical. What differences do you see? Actually, I want to make sure. Yeah, so our chart got split. Yarg. That's fine. How are we doing on time? 43 minutes. We're doing fine. So differences between mitosis and meiosis. We have no DNA duplication in interphase. Of meiosis 2 specifically. G phase may be shortened. That is one that you should probably know. All right, prophase, similarities. Your chromosomes are always going to coil and condense. Spindle always forms, nuclear membrane always breaks down. Those things always happen. In prophase 1, homologous chromosomes synapse. You get synapse. Metaphase, similarities. Something always lines up. We're gonna be precise here. We're gonna say something always lines up on the equator with a metaphase plate. Differences in metaphase in mitosis and in metaphase 2. The 2 here implies that this is meiosis. Individual chromosomes line up, but in metaphase 1, the synapsed pairs line up. All right, anaphase, things are starting to move apart. It moves towards the poles of the cell, opposite poles of the spindle. Anaphase in mitosis and anaphase 2. Anaphase 2. We have our sister chromatids. Separate and move opposite. I'm going to do this in a different color so that we can distinguish it from the box next to it. Sister chromatids separate. Oh, goodness. Separate. There we go. In anaphase 1, members of each homologous pair separate to opposite poles. So in mitosis and anaphase 2, sister chromatids separate. In anaphase 1, members of homologous pairs separate. Finally, in telophase of all of these, the nuclear membrane reforms, the nuclear envelope. And the spindle, which is all of this sort of these microtubules responsible for the structure of this division, breaks down. What's different between them? Chromosomes don't usually uncoil during telophase 1 of meiosis. Just a little further to go. The amount of DNA in a somatic cell is not a gamete, this is all the other cells in the body, equals C during G1 of interphase. How much DNA is present in the cell during each phase of mitosis and meiosis? Mitosis, meiosis, meiosis 2. So in interphase of mitosis, we're going to have C, same number, in G1, and we'll have 2C in G2. Prophase, you'll have 2C in metaphase, 2C in anaphase. And in telophase, you'll have 2C, and it's going to be divided through cytokinesis. And so you'll be reduced back down to C. All right, meiosis 1. Same deal. You're going to have C, 1C in G1, and double that in G2. 1 does not look like a 1. Similar story for meiosis 1. I'm just going to say here C times 2 cells. And in meiosis 2, we stick with 1 times the amount of DNA. So there's no additional multiplication of genetic material in here because the point of meiosis is to get to 1/2 the amount of genetic material from the parent cell. And that just carries across. And you end up with 1/2 the genetic material divided between these 2 cells. How do the similarities in prophase of mitosis and meiosis compare to the similarities in telophase of mitosis and meiosis? So as we talked about before, telophase can be thought of as the opposite of prophase. Opposite. Let's see. The opposite of prophase. In prophase, your chromosomes are consolidating, your nuclear membrane is dissolving, things are starting to move apart. Telophase is quite the opposite. Your chromosomes are starting to uncoil, your nuclear membrane is reforming, and so on. So in other words, whatever's done in prophase is undone in telophase. And finally, at what stages do the most of the differences among mitosis, meiosis 1, and meiosis 2 occur? For what reasons do these differences exist? The primary difference, the most important thing you need to remember, the primary difference between mitosis and meiosis occur as a result of synapsis in prophase 1. So synapsis in prophase 1 let's see, occur as a result of. Let's see. Cause most of the differences. Synapses are what allow the members of homologous pairs to separate to opposite poles, and that reduce the chromosome number at the end of meiosis 1 to half that of the original cell. The synapsis in prophase 1 is what causes most of the difference. Cool. That's everything I've got for y'all. The recording of this video will be uploaded shortly. Be sure and turn in your worksheets for graded completion for that extra credit by Friday. And I will see you all next week.